Hello students, this is Dr. Tikia Arashiro, and in this podcast I'm going to talk about nitrogen fixation, which is a type of inorganic metabolism. The conversion of inert nitrogen gas to a metabolically trackable form such as ammonia is called nitrogen fixation. Nitrogen fixation is a highly oxygen-sensitive process restricted to a select group of diverse microorganisms, often collectively referred to as nitrogen ethers. It is a microbial process in which nitrogen gas is reduced to ammonia. Nitrogen fixation is one of the most important metabolic processes carried out by prokaryotes that are able to fix atmospheric nitrogen to form ammonia. As far as is known, eukaryotes have not evolved this capability, so this process is restricted to prokaryotes. Nitrogen fixation is an energy-intensive process and is an example of an assimilative inorganic metabolism in which the inorganic molecule nitrogen gas is used as a nutrient. The end product, ammonia, which is fixed as organic nitrogen in the cell and used as biosynthetic precursor for the synthesis of nitrogen-containing macromolecules. Nitrogen fixation is inhibited by ammonia. It is inhibited when ammonia is already present and activated when ammonia is absent or limiting. So nitrogen fixation takes place when nitrogen gas is the only major source of nitrogen or when ammonia is absent or limiting. The key enzyme involved in the process of nitrogen fixation is the enzyme nitrogenase. This enzyme is sensitive to oxygen, although it can adapt to low oxygen tension. The key players in nitrogen fixation are nitrogen-fixing prokaryotes, also known as nitrogen fixers. Nitrogen-fixing prokaryotes, or nitrogen fixers, are either free-living nitrogen fixers or symbiotic nitrogen fixers. Free-living nitrogen fixers have no association with a symbiotic partner. They fix nitrogen gas to ammonia on their own. Most phototrophic carbon dioxide fixers are also nitrogen fixers like cyanobacteria, green bacteria, and purple bacteria. Symbiotic nitrogen fixers, on the other hand, are prokaryotes that form symbiotic relationship or associations with other living organisms such as plants and animals. These nitrogen fixers need help with a symbiotic partner to fix nitrogen. The termite gut is a home for a group of important microorganisms. In the gut, you'll find fermenting microbes, methanogens, as well as nitrogen fixers. These groups of microbes have symbiotic relationship with termite and with each other. Now, examples of nitrogen fixers that live in the gut of termites are Citrobacter frondi and Enterobacter agglomerans. Termite takes in wood particles. The fermenters break these wood particles down and produce fermented sugars, carbon dioxide, and hydrogen gas. The methanogens convert the hydrogen gas and carbon dioxide to methane. Some termite species make use of nitrogen fixers to fix atmospheric nitrogen to ammonia to make amino acids. Soybean plants are examples of living organisms with symbiotic relationship with nitrogen fixers. The most common nitrogen fixers are found in the root nodules of soybean plants such as Rhizobium, Bradyrhizobium, and Frankia. So Rhizobium bacteria are nitrogen fixers that live in root nodules of legumes such as soybeans. Rhizobium bacteria infect the roots of soybean plants and stimulate the production of root nodules or swelling of the roots within which they fix nitrogen. These nodules actually result from the invasion of nitrogen-fixing bacteria. If you are to cut a cross-section of the nodules, you will see this densely packed Rhizobium bacteria inside the nodule. Root nodules develop to house nitrogen-fixing bacteria in a microaerobic environment. This process, for the most part, is restricted to a limited number of bacterial groups, including the genera Rhizobium, Bradyrhizobium, and Frankia. A symbiotic relationship is a give-and-take relationship that exists between the soybean plant and the nitrogen fixers. The legumes or the soybean plant benefit from the ammonia produced by the rhizobium bacteria from nitrogen fixation. The soybean plant responds by feeding the rhizobium plant with organic nutrients it makes during photosynthesis. The rhizobium bacteria also receive ATP from the soybean plant to carry out nitrogen fixation as well as organic compounds needed for growth. In the early stages of symbiosis, a complex molecular dialogue takes place involving nod factors synthesized by the bacterium and flavonoids released by legume roots, so that both symbiotic partners can recognize each other and initiate nodulation. Nod gene expression in the nitrogen-fixing bacteria is induced by the presence of flavonoids secreted by legume roots to attract the nitrogen-fixing bacteria, allowing the nod factors to be made. The nod factors then stimulates nodule formation in the legume roots. The nitrogen-fixing bacteria begin to colonize the roots through the infection thread that lead to the formation of nodules. Within these nodules, nitrogen fixation occurs. The reduction of nitrogen gas to ammonia is catalyzed by the enzyme nitrogenase. Nitrogenase can be found in the cytoplasm of nitrogen-fixing organisms. It is repressed when ammonia is already present. Nitrogenase is sensitive to oxygen. Once nitrogen gas is reduced to ammonia, it is fixed in the cell as biomass nitrogen. The enzyme glutamine synthase condenses ammonia and glutamate to glutamine. Glutamate and glutamine are used as biosynthetic precursors for amino groups in nitrogen-containing macromolecules in the cell. So nitrogenase is an enzyme that fixes atmospheric nitrogen or nitrogen gas into ammonia. Although nitrogen gas is abundantly present in the atmosphere, most organisms cannot utilize nitrogen gas directly and must instead take them through other forms like ammonia or nitrate. The triple bond in nitrogen gas is highly resistant to changes in oxidation state, and nitrogenases found in nitrogen-fixing bacteria are the only proteins capable of reducing nitrogen gas to ammonia. Nitrogenase is a dimeric protein, meaning it consists of two proteins, the molybdenum iron protein, also known as dinitrogenase, and the iron sulfur protein, also known as dinitrogenase reductase, which is a ferredox. These two proteins form a complex, and we call the complex nitrogenase enzyme complex. Now, both proteins contain iron centers, and the reduction reaction occurs in the iron. The molybdenum iron protein reduces the proton and nitrogen gas to hydrogen gas and ammonia, while the iron protein receives electrons from ferredoxin, hydrolyzes ATP to ADP and inorganic phosphate, and reduces the molybdenum iron protein. The nitrogen fixation process is an anabolic process and requires 16 moles of ATP to reduce 1 mole of nitrogen gas to 2 moles of ammonia. Nitrogenase catalyzes the following reaction. 1 mole of nitrogen gas plus 8 electrons plus 8 protons plus 16 moles of ATP generates 2 moles of ammonia plus 1 mole of hydrogen gas plus 16 moles of ADP plus 16 moles of inorganic phosphates. So 1 mole of nitrogen gas is reduced to 2 moles of ammonia. 8 proteins is reduced to 1 mole of hydrogen gas, and 16 moles of ATP is reduced to ADP. This reaction is the same as this reaction over here, but this reaction starts with half a mole of nitrogen gas instead of 1 mole of nitrogen gas. So for every half a mole of nitrogen gas, 1 mole of ammonia is generated by nitrogenase. So nitrogenase reaction is a series of reactions during which half a mole of nitrogen gas and 4 moles of hydrogen are reduced to 1 mole of ammonia and half a mole of hydrogen gas. So based on this reaction, the reduction of nitrogen gas to ammonia requires 4 electrons. Let's check the oxidation state table below. The oxidation state of nitrogen gas is 0, and the oxidation state of ammonia is -3. So 0 minus -3 equals 3. So how come it is not 4? 4 electrons are actually consumed in the process. 1 electron is transferred to the molybdenum protein nitrogenase to reduce the proton to hydrogen gas, and the other electrons are used to reduce nitrogen gas to ammonia. So 4 electrons are consumed in the process. 1 electron is lost as hydrogen gas per half a mole of nitrogen gas reduced, and 3 electrons are necessary to reduce nitrogen gas to ammonia. While nitrogenase is sensitive to oxygen, and one might think that nitrogen fixation is restricted to anaerobic microbes because of its sensitivity to oxygen, nitrogen-fixing microorganisms are diverse in terms of respiratory growth. Some nitrogen-fixing organisms are strict anaerobes and can fix nitrogen anaerobically. Some are microaerophilic, so these microbes grow on low oxygen tension. Some nitrogen-fixing organisms are facultative anaerobes and can fix nitrogen when oxygen is absent or present, and some are strict aerobes and can fix nitrogen in the presence of oxygen. Nitrogenases are rapidly inactivated by oxygen, and while they can adapt to low oxygen tension, they need to be protected from oxygen. So what are those mechanisms that nitrogen-fixing microorganisms use to ensure that nitrogenases are active for nitrogen fixation? There are at least 5 mechanisms. The first one is the induction of slime production in nitrogen-fixing cells. So strict aerobes like Azobacter vinylandi produce slime layer on the cell surface. Azobacter vinylandi is a gram-negative, strictly aerobic nitrogen-fixing bacterium. Now, a slime layer is an extracellular material that is formed on the surface of bacteria. The function of the slime layer is to protect the bacterial cells from environmental dangers such as antibiotics and desiccation. Now, slime layer can also serve as a barrier for oxygen diffusion into the cell. So as you can see here, Azobacter vinylandi produces a slime layer on its surface. This slime retards the diffusion of oxygen into the cell, preventing nitrogenase inactivation by oxygen. The colonies of Azobacter vinylandi are often slimy due to the synthesis of exopolysaccharide, which is a major component of slime layers. The second mechanism that microbes have to prevent inactivation of nitrogenase is the ability of some microbes to lower intracellular oxygen concentrations. So some nitrogen fixers are capable of lowering their intracellular oxygen concentration. This mechanism is evident among aerobic Azobacter species. Azobacter species are strictly aerobic organisms and have very active respiratory system. They use oxygen as electron acceptor during respiration. And because they have very active respiration, they apparently utilize oxygen rapidly enough to lower intracellular concentrations of oxygen in the vicinity of the nitrogenase enzyme, allowing nitrogenase activation. The third mechanism is the formation of complex with protective proteins. Some aerobic nitrogen-fixing bacteria, like Azobacter, avoid inactivation of nitrogenase by forming a complex with air-tolerant proteins. These air-tolerant proteins include SHETNA and iron sulfur 2. So this image here displays the nitrogenase enzyme showing the two components, molybdenum iron protein and iron sulfur protein, forming a complex with iron sulfur 2. The complexation of nitrogenase with iron sulfur 2 makes it more tolerant to oxygen, and this prevents its inactivation in aerobic fixing bacteria. As the terminal electron acceptor for aerobic respiration is oxygen, Oxygen stimulates oxidative phosphorylation, and it provides ATP supply to fuel both nitrogenase activity and its synthesis. Recall that nitrogen fixation is an energy-intensive process and requires 6 moles of ATP to reduce 1 mole of nitrogen gas to 2 moles of ammonia. So it relies on aerobic respiration for the supply of ATP to fuel its activity. The optimum oxygen concentration for maximal nitrogen fixation rates varies widely among nitrogen-fixing organisms, with some showing considerably more oxygen tolerance than others. In vivo, nitrogenase is not only a potential target for inactivation of oxygen, but also be a source of superoxide-derived radicals that can further the cellular superoxide-related damage. For example, under certain oxygen conditions, the iron-sulfur protein of nitrogenase can reduce oxygen in a reaction that produces either hydrogen peroxide or superoxide as an intermediate. So some aerobic nitrogen-fixing bacteria utilize nitrogenase protective proteins, such as Shetna and iron-sulfur, to prevent nitrogenase inactivation by oxygen. The fourth mechanism is the compartmentalization of nitrogenase in heterocysts. Filamentous cyanobacteria, such as Anabaena and Nostoc, compartmentalize nitrogenase in a heterocyst to protect the enzyme from oxygen deactivation. Filamentous cyanobacteria protect nitrogenase by differentiating 5 to 10% of its vegetative cells into special nitrogen-fixing cells called heterocysts when ammonia is absent. Cyanobacteria are oxygenic photoautotrophs, and oxygenic phototrophs are nitrogen fixers and also carbon fixers. So they fix nitrogen gas and carbon dioxide in the atmosphere and convert them to sugar during photosynthesis. The oxygenic photosynthesis occurs in the vegetative cells, while the nitrogen fixation occurs in the heterocysts. So cyanobacteria are able to compartmentalize nitrogenase in the heterocyst to prevent it from being exposed to oxygen and be inactivated. Recall that oxygenic photosynthesis results in the production of carbohydrates and oxygen inactivates nitrogenase. So a mechanism that cyanobacteria use to prevent this from happening is by producing heterocysts. So heterocysts are formed by some filamentous cyanobacteria like Anabaena and Nostoc when ammonia is limiting or absent. So when ammonia is limiting or depleted, some vegetative cells are differentiated into heterocysts. Now, cell differentiation is a process where cell changes from one form to another. So an example of cell differentiation is when some vegetative cells evolve to become heterocysts. It is important to remember that heterocyst formation only occurs during nitrogen starvation or when ammonia is limiting or depleted. In a scenario where ammonia is plentiful, no heterocyst is formed. So when ammonia is limiting, cyanobacteria fix nitrogen in specialized cells called heterocysts and carry out oxygenic photosynthesis in the vegetative cells. Heterocysts vary from vegetative cells in many respects. Heterocysts are the site of nitrogen fixation, and they express the enzyme nitrogenase, while vegetative cells cannot express nitrogenase. Heterocysts do not fix carbon dioxide, while vegetative cells do. They fix carbon dioxide to form carbohydrate via the Calvin cycle. Some of the organic carbon enter the heterocyst to serve as carbon source for the synthesis of glutamine and glutamate. Heterocysts do not produce oxygen, so they lack oxygen-producing ability, while vegetative cells can produce oxygen through oxygenic photosynthesis. Now, oxygenic photosynthesis occurs in the vegetative cells, and one of the end products of oxygenic photosynthesis is oxygen. Heterocysts are surrounded by a thick cell wall containing glycolipid and polysaccharide, which are believed to serve as a permeability barrier to atmospheric oxygen. So the cell walls of heterocysts are airtight while the vegetative cell walls are permeable to oxygen. Finally, heterocysts do not divide. They are not capable of cell division, while vegetative cells are capable of cell division. So filamentous cyanobacteria, such as nostoc and anabaena, grow by division of all of its vegetative cells. The heterocysts do not divide. Now, the fifth mechanism is oxygen protection by the symbiotic partner. Recall that some nitrogen-fixing bacteria are free-living and can fix nitrogen on their own, while some rely on a symbiotic partner. Now, Rhizobium is one of those. So how does the symbiotic nitrogen-fixing bacteria protect nitrogenase from oxygen inactivation? In plants infected with Rhizobium, the presence of oxygen in their root nodules would reduce the activity of the oxygen-sensitive nitrogenase, which is responsible for nitrogen fixation. Oxygen concentration is controlled in the outer cortex of the nodule. In the outer cortex, you will find the leghemoglobin protein. This protein has high affinity to oxygen. It is an oxygen carrier, and it lowers the oxygen concentration in the inner cortex. It buffers the concentration of oxygen in the inner cortex low enough to allow nitrogenase to function, but high enough to provide the Rhizobium bacteria with oxygen for respiration. That's the end of the third podcast on inorganic metabolism. Stay tuned for the last podcast.